Welcome to my first podcast. This is on chapter 14, Mendel and the Gene Idea. In these podcasts, I'm not going to cover all the material that I cover in the lectures, but rather focus in on each of the concepts. In concept 1, we discuss how Mendel used a scientific approach to identify the two laws of inheritance. Mendel was different than his contemporaries. He was a true experimentalist. There are two key points. He followed multiple generations in his experiments, and he also took a quantitative approach, setting up literally thousands of these breeding crosses. He was also quite lucky. He picked peas for his experiments, and they have the advantage in that they're self-fertilizing, but that he could also set up crosses. So here's a cross between a white plant and a purple plant. Here's that cross shown in a different way, and it makes the point that Mendel tracked heritable characters for 3 generations. So the first generation is the parental generation. These are true breeding parents, so the purple flowered plants crossed to the white flowered plants to produce the first filial, or the F1 generation, and all these hybrids had purple flowers. He then allowed these plants to self-pollinate to produce the F2, or the second filial generation, and here now you see this famous 3-to-1 ratio emerge. This monohybrid cross, which is just involving a single gene, 2 different alleles of the same gene, allowed Mendel to propose the law of segregation. So here's that same cross now written out with the genotypes given. So the purple flowers are true breeding, big P, homozygotes. The white flowers are also true breeding homozygotes, but in this case they carry the 2 recessive alleles, little p. In the first filial generation, you produce heterozygous plants, big P, little p. The third generation that we're considering, the F2 generation, we're actually seeing how they are formed using what's called a Punnett square. And a Punnett square is a very simple way to determine how the gametes are going to combine. So in this generation, there are 2 kinds of gametes produced, big P and little p. So if we write these out from the sperm and the eggs, and then combine them randomly in this Punnett square, you'll see the famous 3-to-1 ratio. So if we look at the genotypes of these plants, this plant is big P, a homozygote. There are 2 heterozygotes, but because big P is a dominant allele, both of these are purple. And this fourth one is little p, and it's the white flowered plant. So this monohybrid cross allowed Mendel to propose the law of segregation, which states that allele pairs, or versions of the same gene, different alleles, are going to separate or segregate during gamete formation, and then they're going to randomly combine at fertilization, and that's what we saw in that Punnett square. It took a different kind of cross for Mendel to propose the law of independent assortment, and this is called a dihybrid cross because it actually involves 2 traits, so alleles for 2 different genes, big Y, big R, little r. So here are the parents. So this parent produces peas that have yellow round peas, and this plant produces peas that are green and wrinkled, and here are their genotypes. Again, they're both homozygous true breeding strains. They're crossed together to produce the F1 generation. These are the double heterozygotes. So if you were to propose a hypothesis of dependent assortment and draw out the corresponding Punnett square, this dependent assortment states that big Y, big R are going to stay together in the gametes, as are little y, little r. So if you draw out the Punnett square, you can immediately put a big line through it, because these predicted phenotypic ratios did not fit with Mendel's observed results. His observed results rather fitted with a hypothesis of independent assortment, and this states that in this heterozygote, the gametes that are produced with alleles from these different genes combine independently. So with equal probability, you form a gamete that has big Y, big R, big Y, little y, big R, little y, little r, and all of these gametes form at an equal probability. So Mendel's law of independent assortment, derived from this dihybrid cross, states that allele pairs from different genes separate during the formation of gametes and then assort independently with alleles from each of these genes. So traits are transmitted to offspring independently of one another. And this holds for genes that are on different chromosomes, which was the case actually for most of the genes that Mendel studied in his experiments, and also for genes that are widely separated on the same chromosome. And we'll consider this second point later. The second concept is a way to help you understand how to do genetic problems, and that is that the laws of probability govern patterns of Mendelian inheritance. And the analogy that we can use in thinking about these problems is that of a coin toss. So here's a coin, heads or tails, and just as you can have two alleles for a single gene, big R, little r, we've labeled the size of the coin in the same way. Two rules apply. The rule of multiplication. So here's the question. What's the chance that two coin tosses will produce either two heads or two tails? The chance of producing heads is a half. It can either be heads or tails. The chance that the next toss of the coin also produces a head is also a half. So the chance that we'll get two heads in a row is just multiplying those two probabilities together and to give you a quarter. So we can restate that question genetically and ask, what is the chance that an F2 individual will be homozygous, either big R or little r? And again, let's look at the Punnett square here to see these genotypes. So it's exactly the same idea, and the chance that you'll be big R is a quarter. There's another rule, and that's the rule of addition, and we use that to answer the following question. What's the chance of an F2 individual being heterozygous? And if you look in the Punnett square, there are actually two ways that you can be heterozygous. So to determine the chance that any given F2 individual will be heterozygous, we have to add together those two probabilities, a quarter and a quarter, and the answer is half. The third concept is that everything's not quite so simple. So we have to consider examples where the inheritance patterns at first view don't appear to be predicted by simple Mendelian genetics. And we're going to look at three ways to extend Mendelian genetics when just considering a single gene. And the first of these is when alleles are not completely dominant or recessive, and that's the phenomenon called incomplete dominance. So in this example, there are two alleles, CR and CW, and here is snapdragon plant. So this is the homozygous plant having the CR allele, and you can see it's fully red, and it's crossed to a plant that's homozygous for the C white allele. In the F1 generation, the heterozygotes are pink. So what this tells you is that possession of the CR allele is not fully dominant, and the plants appear pink. If we now follow this to the next generation, so cross these plants together, now you'll see a red plant appearing in the F2 progeny, and this F2 has two copies of the CR allele. So this phenomenon where possession of this allele is not fully dominant is called incomplete dominance. Another example where you don't see simple Mendelian inheritance patterns concerns blood type. So this is a very well-known example, and there are actually multiple alleles that give you your blood type. And in the figure you can see here these blood types A, B, and A, B, and O. And if you're an A blood type, then you have a certain pattern of carbohydrates on your red blood cells. If you're B, you have a different pattern. But if you have these two alleles, both A and B, and here's the genotype over here, then you actually have both kinds of carbohydrates. So they're codominant, you see both types. And of course there's blood type O, and in these cases your genotype is i, and there are actually no carbohydrates on the red blood cells. So these are multiple alleles and they show codominance. There's another phenomenon called pleiotropy, and this considers the idea that most genes actually have multiple roles in biology. So when a gene's defective, you see lots of different phenotypic effects, and this phenomenon is called pleiotropy. In terms of certain hereditary diseases such as cystic fibrosis and sickle cell disease, many functions are disrupted, so you'll see multiple organs involved in this disease. And in the figure, there's a chick, so here's the normal one on this side. This is a mutant for a single gene, but you can see it has a phenotype involving all different kinds of aspects. So this chick has no feathers, no lungs, its toes are short. So it has a pleiotropic effect because this gene's actually involved in many different biological processes. We can also extend Mendelian genetics for 2 or more genes, and we're going to consider 2 examples of this. The first one is called epistasis, and this is a cross, in this case, between 2 mice, and we're looking at 2 different genes, so a gene involving coat color and whether pigments are put into the fur. So this mouse, these are 2 heterozygotes, they cross together, and here's the Punnett square, and we see an unusual ratio. So let's look a little bit about these genes. So B is for black coat color, so it's a dominant allele. So if you're either double homozygous big B, or big B, little b, you'll have a black fur. And if you're little b, you'll have brown fur. And we're also looking at another gene. So let's look at this mouse here in the Punnett square. According to its genotype at the B locus, it should have black fur, but it's white, as you can see in this figure. So let's look now at C. So in this case, this mouse is homozygous recessive for the little C allele, and if you're homozygous recessive, then no pigment is deposited into your fur. So even though the pigment's being made, the mouse looks white because its genotype at this C locus masks the phenotype of the other. And this masking of one phenotype by another is called epistasis. Another example is polygenic inheritance. So many characters, many attributes of our phenotypes are not conferred by a single gene. It often involves 2 or more genes, and a common example of this would be skin color. So in the example, we're supposing that there are 3 genes that confer differences in skin color, and this huge Punnett square and the different combinations of these different genes shows how in this individual you can go from very dark skin color all the way to this individual who has very pale skin color, because this trait of skin color has been conferred by multiple genes. It's a polygenic trait. And then in the final concept, we think about how Mendelian patterns of inheritance apply to human genetics. We're not very good subjects for genetic research for lots of different reasons, but basic Mendelian genetics applies, and it's sort of done retrospectively in humans. So we look at pedigrees, we look at family trees. You can recognize traits that are dominant and recessive. And if we think about this just for disease genes, so in a dominant disease gene, heterozygotes who possess that allele will express the trait, and they can expect that about half of their offspring will also be affected. However, if a disease gene is recessive, then you need to be homozygous to have the disease or express the trait. And for disease genes, then the parents must be carriers or heterozygotes, and they can expect about a quarter of their progeny are going to show the trait. So in a lecture, we're going to consider a lot of different examples of human genetics. So let me just close by saying that it's important for you to know that to learn this material, you shouldn't rely on this podcast. It's a very quick summary. You should read the chapter, come to lecture, and then go and test your knowledge. So thank you very much for listening to my first podcast.